Thank you.
Thank you. Okay. Okay, thank you, Dr. Cui, for the very warm and encouraging opening notes. So now let's get into our main oral presentation session. And in this session, each finalist will have about 22 minutes for oral presentation and about 8 minutes for Q&A with our committee members. So now, please welcome our very first finalist, Dr. Jiamu Du, from Southern University of Science and Technology in China. And he's going to tell us about his research on plant DNA methylation. Dr. Du, are you ready?
Is that okay now?
We haven't seen your, oh, yes, it's working.
It seems to have a little delay. Okay.
Yeah,
so
it's working now.
May I start?
Yes, please.
Okay. Thank you, Wenjia. Hello, everyone. It's my great honor to be selected as one of the finalists of this RSPS 2023 program. And I'm very happy to have this opportunity to share the work progress in my lab. So today, I will mainly talk about our recent work progress about the molecular basis of the establishment, maintenance, and removal of DNA methylation in plant. Okay, so first, it looks okay. So first I would like to have a very brief introduction of the background. So in eukaryotic cells, including plant cells, besides the classic DNA sequence-dependent gene regulation, there are extensive DNA sequence-independent gene regulations, including DNA methylation, histone non-coding RNA, and chromatin structures. So these are classified as epigenetic regulation. So in my lab, we mainly work on the structure and the biochemical studies of the molecular basis underlying plant epigenetic regulation with a focus of histone modification and DNA methylation. So today, I will mainly talk about our recent works on DNA methylation in plants. So biochemically, DNA methylation is a simple chemical reaction that adds a methyl group to the fifth position of the cytosine base to produce this 5-methyl cytosine, 5-MC. Genetically, the 5-MC indicates the gene silencing signal and conducts the gene silencing related functions such as genome imprinting and heterochromatin generation. So in plant, DNA methylation is exceptionally important because we know that the plant always have a much bigger genome than animals. This doesn't mean plants have so many genes, but rather they have much more transportable element and repeat sequence within their genome. And this element may make the plant genome very unstable. Then plant require a very strong DNA methylation system to systematically suppress this transportable element and repeat sequence enriched in their genome. And also this type of suppression needed to be maintained in the next generation during DNA replication. Therefore, the regulation of DNA methylation can be biochemically divided into three different steps. So first is the establishment of DNA methylation pattern from unmethylated DNA, and this process is called de novo establishment of DNA methylation. We know that during DNA replication, the newly synthesized DNA is totally unmethylated. So after DNA replication, the plant needed to recover the DNA methylation, which is guided by the pre-existing DNA methylation patterns. So this process is called the maintenance DNA methylation. While in some certain conditions, plant also needed to actively remove this DNA methylation to recover it back to the regular cytosine to rescue the gene activity. So this process is plant DNA demethylation. So in my lab, we systematically worked on all these the biochemical basis of all these three processes controlling demethylation in plants. Let's start. Let's start with the de novo demethylation first. So in plant, all the de novo demethylation is conducted by a plant-specific pathway called RNA-directed demethylation, RDDM. So in this pathway, there are two plant-specific RNA polymers, Pol V and Pol V. While the Pol V produce single-stranded RNA, which is finally processed into 24 nucleotide siRNA, the Pol V directly produces long non-coding RNA. Once this Pol V produced 24 nucleotide siRNA base pair with this Pol V produced long non-coding RNA, they can initiate the gene silencing signal and transmit this signal to the downstream demethyltransferase DRM2 to facilitate local specific demethylation. So in the upstream of RNA-directed demethylation, a key question is that how this 24 nucleotide siRNA is produced. So we know that in eukaryotic cells, the length-specific small RNA production requires a group of ribonucleases called dicer. In the RDDM pathway, plant uses dicer3 enzyme to produce this siRNA in a length of 24 nucleotides. But we know that the dicer only work on the double-stranded RNA, while the RNA polymerase only produce single-stranded RNA. So plant need one more step of this plant-specific enzyme called RNA-dependent RNA polymerase 2, RDR2, to convert the single-stranded RNA into double-stranded. Then feed this RNA to the dicer3 for the next step of dicing. To investigate the molecular basis of this process, we carried out our structure studies. So our structure study suggests that the K enzyme RDR2 in absence of the RNA, the K enzyme RDR2 adopts a closed conformation, which here we can see the active site is deeply embedded inside this protein, which suggests that the enzyme is in an inactive form. Once the Pol V produced single-strand RNA goes into this active center, it can trigger the conformational change of the RDR2 to open this RNA channel and then to convert the enzyme into active form. For the efficient double-stranded RNA production for the next step of dicing. So in the next step, we successfully captured the active dicing conformation structure of dicer3 in contact with its substrate RNA. And with this structure, we revealed the molecular basis for the terminally specific RNA recognition by the dicer3, and more importantly, we revealed the molecular basis for the 24 nucleotide length measurement maximum for the dicer3. And this is also the first dicer RNA active conformation complex. So our structure also revealed the length measurement and the dicing principles of dicer in general. So after this upstream work, let's move to the downstream. So the key component of the downstream of RDDM is the RNA polymerase 5. And RNA polymerase, we know that the biochemical function of Pol V, the biochemical function of RNA polymerase of RNA polymerase 5 is forward on the chromatin to continuously produce this RNA transcript. So it's needed to go on the chromatin. While physiologically, the physiological function of Pol V indeed is a simple chromatin locator to stand on the chromatin and tell the downstream DNA methyltransferase DRM2 which site needed to be methylated. So it is not to go. So there is a contradiction between the biochemical to-go function of Pol V and the physiological not-to-go function of Pol V. Therefore, there must be a mechanism that modulates the biochemical function of Pol V to make it adapted to its physiological function. So to investigate this mechanism, we start our Pol V. Study. So we know that Pol V is derived from Pol II. So it contains 12. It is a bigger complex with 12 subunits, and moreover, Pol V is located at the RDDM pathway, which is very upstream of gene regulation. So the endogenous abundance of Pol V is quite low. So a key challenge in this project is how can we obtain enough Pol V sample for our structural study. So we worked on this, we worked very hard on this project for several years. Once a collaborator told us that in the early years, plant biologists always directly purify proteins from the cauliflower. Then I suddenly know what the key is in this project. So we obtained an antibody against the cauliflower Pol V, and I asked 3 of my students to form a team. And I told the team that the task of your team is very simple. So you guys can go, every morning you guys go to market and buy 10 kilograms of cauliflower and then try to purify the Pol V from the cauliflower. But you know that although we know the Pol V is there, the purification is still very challenging. So it happened quite common that in the morning the students came to my office said Dr. Du, we are happy that we saw our Pol V in our sample, but 4 hours later in afternoon they came to my office again and said we don't know what happened, but indeed we cannot find our Pol V anymore. So this happens again and again. So during past 5 years, we used more than 1.5 tons of cauliflower, and then finally we established a successful protocol for the large amount of purification of Pol V. So with this purified Pol V, we successfully determined the pro-EM structure of the plant Pol V in its elongation conformation. So generally, the overall structure of Pol V is quite similar at Pol II. So I will not discuss it in detail. I will just only focus on how Pol V is adapted to its function. We know that the general working procedure for the RNA polymers is that the upcoming double-stranded DNA goes into the RNA polymerase, and then the double-stranded DNA breaks into two strands. One strand is a template strand goes into the active center and to serve as a template for the RNA synthesis. While the other strand is a non-template strand, usually it is looped out, and 10 nucleotides later, these 2 DNA strands will anneal together. So in our structure, we have a very interesting observation that a tyrosine residue specifically stacks at the double-stranded DNA branch site to abolish the DNA forwarding into the active site. So this tyrosine is Pol V conserved, but not conserved in Pol I, Pol II, Pol III. So we can have a conclusion that this Pol V conserved tyrosine residue can prevent the double-stranded DNA going forward to the active center, and then it can slow down the overall transcription rate. And another key feature for the RNA polymers is that it is not only it does not always going forward, but sometimes RNA polymerase often goes backward to self-check for the very essential proofreading function. So it is very interesting that in our in vitro assay, we observed a very strong backtracking induced 3' to 5' cleavage activity of Pol V compared with Pol 2, which means that Pol 5 have an enhanced backtracking activity. So we want to know where this enhanced backtracking activity comes from. So as I just mentioned, that the RNA polymerase in RNA polymerase, the non-terminal stranded DNA usually is looked out. But in our structure, we notice that the basis of the non-terminal stranded DNA is specifically captured by several surface pockets of the NRP2 subunit of Pol V, which is a Pol V specific subunit. So when we mutate the NRP2 bonded non-terminal stranded DNA bases into AP site, then in the assay, in our in vitro assay, we observed a comparable transcription elongation, but a much reduced backtracking activity. So this means that the Pol V used its NRP2 subunit to enhance the backtracking of the transcription. Then overall, our study revealed that the Pol V used its unique tyrosine residue to slow down the transcription and use its NRP2 subunit to enhance its backtracking. When these two effects come together, this can make the Pol V hesitate to move forward, but rather stand there to enhance the chromatin retention for the efficient DNA methylation. After revealing the mechanism for the de novo DNA methylation, let's move forward to the maintenance DNA methylation. So indeed, about 10 years before, when I was a postdoc, my work had already revealed the molecular basis for the maintenance DNA methylation implant. The histone S3K9 demethylation can direct the DNA methyltransferase 72, 73, and even RDDM to deposit the DNA methylation, while methylated DNA can direct the switch family histone methyltransferase to deposit the histone S3K9 demethylation. So implant this histone methylation directs the DNA methylation, and the DNA methylation directs the histone methylation can form a positive feedback loop. Then this positive feedback loop can faithfully maintain the DNA methylation implant. Therefore, a key step of the maintenance DNA methylation is to create a suitable histone modification state of histone S3K9 demethylation as well as the histone S3K4 unmethylation. So in my lab, we further studied how this plant to use the demethylated domain defaulting to specifically remove the K4 methylation, and how this methylated DNA can direct the histone methyltransferase switch 6 to deposit the methylated histone S3K9. This positive feedback loop can faithfully maintain the plant DNA methylation, but it also introduced a serious problem because we know that the positive feedback loop is always out of control. So this DNA methylation can be maintained by this positive feedback loop, but it may also extend it to all over the genome to silent averaging. So plant also requires a strong DNA demethylation system to balance this DNA methylation system. So this is the ROS1-based DNA demethylation system in plant. So in animal, the DNA demethylation is much complex, that it uses the tight enzyme to oxidize the 5-MC base and use the second enzyme TDD to actually remove this base to produce this AP site. for the downstream DNA repair to fill this site with regular cytosine to achieve the DNA demethylation. But in plant, a single enzyme ROS1 or its homologous can actively exceed this 5-MC base and insert the sugar ring to create this single-stranded DNA break for the downstream DNA demethylation. So plants have a much more robust and simple DNA demethylation system. However, although ROS1 was identified 20 years before, it is still a big challenge for our structure study because the keyword of ROS1 is discontinuous. So we took this challenge because ROS1 have two very long intrinsic disordered regions. So in about 8 years, we tested hundreds of constructs, and finally we obtained a shortest construct of ROS1, which removed most of the disordered region but kept its activity. So with this construct, we successfully determined the structure of ROS1 in contact with its substrate, the DNA. So overall, ROS1 adopts a classic DNA repair-related DNA glycosylase lipase bifunction enzyme-like fold. The 5-MC is flip out and specifically recognized by ROS1. So we can make a conclusion that the DNA repair enzyme-derived ROS1 can treat the 5-MC as a DNA damage and then directly repair this 5-MC DNA damage to achieve the DNA demethylation. Finally, I would like to have a summary that in the past years, we systematically studied all the 3 steps that regulate plant demethylation and reviewed the biochemical and structural basis for all these processes. Finally, I would like to thank all my collaborators in all these projects, and thanks for your attention.
Okay, thank you, Dr. Du. A very nice talk. So now it's time for our Q&A section with the committee members. So for all the committee members in the chatting room, if you do have questions for Dr. Du, please directly turn on the microphone. Yeah, please go ahead.
So maybe I will ask the first question. So Jiamu, very nice talk. So you mentioned the 24 nucleotide siRNA is produced by DASA 3. As we know, there's several DASAs encoded by the plant genomes. So I'm curious, what's the difference between the different DASA proteins? So that make them to produce different nucleotide or small RNA. So what's the difference between different DASA?
Okay, so first, different DASA locates in different pathways. For example, the DASA1 produces microRNA, so it recognizes the bubble of the precursor microRNA. So the DASA 2, 3, 4, they all produce this small interfering RNA. So their substrate are the perfectly base paired double-stranded RNA. And for the major difference is that first they have different terminal specificity. For example, DASA 3 prefer the guided strand with A in located at the first base, but others have different preferences, and their length measurement is different. For example, DASA 3 measures 24 nucleotide, while DASA 2 measures 22, and DASA 1 and DASA 4 measure 21 nucleotides. So their length measurement are different.
Okay, is there a structural basis? For example, there's a specific domain or motif that can recognize different lengths of the double-strand RNA. Is there any structural basis?
Indeed, we are working on this now. So, so far as I know, I think the principle is quite similar because we know that we can imagine the double-stranded RNA. We can think it like a long stick. So the dicer with one domain covers on one terminal, and then if it wants to cut at 24, then the protein length is about is just for the 24 nucleotide length. So I think for the measurement, it's just adjust the relatively position between the PEZ domain and the ribonucleus domain, so they can measure different lengths and cut it.
Okay, I see. Thank you, Jiamu.
Okay. Thank you.
Right. Hello, Jiamu. Very nice talk.
Okay, thank you.
Yeah, I just we just know the dynamic change of demethylation or demethylation is very important for gene expression. In your structure, did you see any amino acid modification in your structure, which may indicate some regulation of this progress?
You mean protein modification? Or...
Yes, in a structure. Yes, some specific amino acid modification. Yeah.
Currently, no. So because most of the protein are expressed from E. coli or the insect cell, so the modification system is different from the plant cell. So we don't know if there is any modification can regulate the DNA methylation or DNA demethylation. But as I know, in animals, the DNA methyltransferase itself can be methylated. So this is one more step for the methylation of DNA methyltransferase regulated DNA methylation. So it happens in animal, but in plant, so far I didn't notice there is any report about this.
Okay, thank you.
Thank you.
Okay, we still have time for one more question. Any committee members that have a question?
Maybe I have a very general one. So I'm absolutely not a specialist here, but I read in your proposal also that you were pointing to the plant-specific features of this epigenetic regulation systems, and you relate that to the unique life cycle and the unique life strategy that plants have to adapt to, probably to environment. And so could you just comment on this? What can you see in the plant epigenetic regulation systems that would be helpful for them to survive in a changing environment, in a more dynamic environment as opposed to animals, or it's just a very general question?
Okay, thank you. So for this question, so my feeling is that for the rapid response, I think the key will be the histone modification. So this is another area we are working on, but I didn't include it in my application. For the long-term response to environmental signals, I think the DNA methylation must be involved in, because for the long-term adaptation to the environmental signal, so there must be some genome level genome level change, so the demethylation can systematically or in a long range to silence a fragment for the genome. But for the quick response, I believe it must be the histone modification.
Okay, thank you.
Thank you.
Okay, thank you again, Dr. Du, for